After discussing about gram positive and gram negative types of bacteria, now let us discuss about nutrition in bacteria. Nutrition in bacteria. Nutrition in bacteria. So nutrition in bacteria, for easy understanding, we can broadly divide it into two types. We can broadly divide it into two types. One is autotrophic. One is autotrophic. Another one is heterotrophic. One is autotrophic, another one is heterotrophic. So autotrophic means can synthesize their own food material. Can synthesize their own food material are called as autotrophic nutrition. Okay, is called as autotrophic nutrition. Heterotrophic means cannot synthesize their own food material. Okay, cannot synthesize their own food material, but depends on others. So such type of nutrition is called as heterotrophic. Autotrophic can synthesize their own food material. Heterotrophic cannot synthesize their own food material but depends upon others. Okay, so this autotrophic nutrition in bacteria is again classified into two types. One is photoautotrophic. Another one is chemoautotrophic. Photoautotrophic, another one is chemoautotrophic. Okay, photoautotrophic means here chlorophyll is present. Okay, due to the presence of chlorophyll, the bacteria undergoes photosynthesis and prepare its own food material. So such are by using the light, okay, are called as photoautotrophic. Example chlorobium. Okay, chlorobium, chromacium, rhodospirillum. Okay, chorobium, chromacium and rhodospirillum are examples of photoautotrophic bacteria. Okay, next chemoautotrophic bacteria. So chemoautotrophic means here non-chlorophyllous bacteria. Okay, non-chlorophyllous bacteria, they prepare food by oxidizes. Okay, by oxidizing. Okay, so here chemoautotrophic means non-chlorophyllous and they prepare their food. Okay, they prepare their food by oxidizing, okay, by oxidizing or oxidizes inorganic substances, by oxidizing or oxidizes inorganic substances. Okay, so example, the best example, okay, the best example is nitrifying bacteria. Okay, they prepare their own food materials by oxidizing inorganic substances. Example, nitrifying bacteria. How they'll oxidize? NH4 is oxidized. Okay, NH4 is oxidized by this nitrifying bacteria into 2 minus NO3, NO2, NO3. NO2 and NO3. Okay, so NO2 by nitrifying bacteria converts NH4 ammonia into NO2 and NO3, nitrite and nitrate and nitrate. For conversion, here conversion takes place with the help of, okay, the conversion takes place with the help of bacteria nitrosomonas. Okay, nitrosomonas. Here nitrobacter, nitrococcus. Okay, here nitrosomonas bacteria, nitrobacter and nitrococcus. Okay, so these are the examples, best examples of chemoautotrophic bacteria. Okay, next, coming to the heterotrophic. Heterotrophic bacteria are classified into mainly three types. They are parasitic, saprophytic and symbiotic. Parasitic, saprophytic and symbiotic. Parasitic means depends on host. Okay, depends on host. Saprophytic means depends on dead and decaying organisms. Depends on dead and decaying organisms. So here association, bacteria shows association with other organisms. Bacteria shows association with other organisms, such are called as symbiotic. Parasitic, saprophytic and symbiotic. Parasitic means bacteria that depends upon host. Saprophytic means depends upon dead and decaying organisms. Symbiotic means shows association with the other organisms. Okay, so now this parasitic is again divided into two types. This parasitic is again divided into two types. Parasitic is again divided into two types. One is obligatory parasite, another one is facultative parasite. Obligatory parasite, another one is facultative parasite. So what is meant by obligatory and facultative parasite? Obligatory parasite means completely depends on host. Completely depends on host. If host is not available, then bacteria will die. Okay, then bacteria will die. That means obligatory parasite means completely depends upon the host. Without the host, they cannot survive. Facultative means they are generally parasitic. Generally they are parasitic. But if host is not available, then they live as saprophytes. Facultative parasitic means generally they are parasitic. That means parasitic means they depend upon the host. Even though if host is not available, then this bacteria will live as saprophytes. Okay, then they live as saprophytes. Such type of parasitic bacteria are called as facultative parasites. Such bacteria are called as facultative parasites. Again, saprophytic. This is also divided into two types. Obligatory saprophytic, facultative. Saprophytic. So obligatory saprophytic means here they are completely saprophytic. They are completely saprophytic. That means completely they will depend upon dead and decaying organisms. Okay, they are called as obligatory saprophytes. Facultative means here on reverse. They are generally saprophytic, but when dead and decaying organisms are not available, they live as parasitic. Okay, live as parasitic when dead and decaying organisms are not available. When dead and decaying organisms are not available. Okay, so those are called as facultative saprophytic. So obligatory parasitic example: Microbacterium leprae, which causes leprosy disease in man. Here facultative Example, Microbacterium tuberculosis. Microbacterium tuberculosis, which causes TB disease. Okay, so example. Next here, saprophytic. Example, Bacillus vulgaris. Okay, Bacillus vulgaris. Facultative saprophytic. Example, Pseudomonas. Pseudomonas. Okay, Pseudomonas. Okay, so these are about, this is about parasitic and saprophytic. And last one is the symbiotic. Last one is the symbiotic. Last one, symbiotic. Symbiotic means shows association with others where two organisms are benefited. Where two organisms are benefited. Shows association, okay, with other organisms where the two organisms are benefited. Okay, so the symbiotic for the symbiotic examples in legume plants. In legume plants, example, Rhizobium bacteria. Rhizobium bacteria shows, this Rhizobium bacteria shows association with legume plants where Rhizobium bacteria gets food and shelter from legume plants, and in reverse legume plants gets nitrogen through the nitrogen fixation process from this bacteria. So this is in legume plants. In non-legume plants, okay, in non-legume plants, example, in non-legume plants, example is the Frankia. Example is the Frankia. Okay, non-legume plants, example is the Frankia. Okay, non-legume plants may be Casuarina, Aulus Mirica. Okay, so in this Frankia bacteria shows association. So this is about the complete nutrition in a bacteria, how the basing on the nutrition, how the bacteria are classified. Okay, so this is about the nutrition in a bacteria. Next topic, next one is the reproduction. Reproduction in bacteria. So first mode of reproduction is vegetative reproduction. Generally in bacteria, vegetative reproduction takes place, okay, takes place by budding, and by binary fission. Okay, takes place by budding and binary fission. Vegetative reproduction takes place by budding and binary fission. Next, second method, by asexual reproduction. Asexual reproduction takes place by endospores. Okay, these are thick spores, okay, which are resistant to heat and temperature. Generally the bacteria forms thick spores called as endospores, which are resistant to heat and temperature. So they are resistant, and the bacteria the endospores can survive in high heat and high temperature conditions. When the favorable conditions are available, these endospores now develop into a new individual bacteria. Okay, so that is about endospores. Next, okay, so here another important point is these are highly resistant due to the presence of calcium diplocolinic acid. Due to the presence of calcium diplocolinic acid, so their endospores are resistant to high temperature and heat. Okay, next, second one is by zoospores. By the formation of asexual reproduction takes place by the formation of zoospores. Asexual reproduction takes place by the formation of zoospores. Example rhizobium species, and asexual reproduction also takes place by conidia. Okay, by the formation of conidia. Example filamentous bacteria like Frankia. So these are the asexual reproduction takes place by the endospores, zoospores, and by the formation of conidia. Also, here the asexual reproduction takes place in a bacteria. Next, third one is sexual reproduction or genetic material transfer. Sexual reproduction, or it is called as genetic material transfer. Okay, genetic material transfer. So how this takes place? How this sexual reproduction or genetic material transfer takes place in a bacteria? So here sexual reproduction occurs in bacteria by three methods called as transformation, conjugation and transduction. Okay, sexual reproduction occurs by three methods called as transformation, conjugation and transduction. Transformation, first one is the transformation. So transformation means here genetic material is transferred by unknown mechanism. Okay, transformation of genetic material takes place by unknown mechanism is called as transformation. First discovered by Griffith. First discovered by the scientist Griffith. Okay, genetic material is transferred by unknown mechanism, first discovered by Griffith. In Streptococcus pneumonia. So genetic material is transferred by unknown mechanism, first discovered by Griffith in Streptococcus pneumonia. It is also called as Griffith effect. Okay, also called as Griffith effect. Next, second one is conjugation. Second one is conjugation. Here conjugation means transfer of genetic material. Okay, transfer of genetic material from one to other bacteria through a conjugation tube is called as conjugation. Okay, is called as conjugation. Transfer of genetic material from one bacteria to other bacteria through a conjugation tube. So here the diagrammatic representation, if you observe, here a conjugation tube is formed, and this genetic material is transferred from F plus bacteria to F minus bacteria. Okay, from F plus bacteria to F minus bacteria, the genetic material gets transferred through this conjugation, through the conjugation tube. Okay, so first discovered, this was discovered discovered by Liderberg and Tatum. Liderberg and Tatum in E. coli. In E. coli bacteria. In E. coli bacteria. In E. coli bacteria. Next, third one. Third method is transduction. Here, transfer of genetic material occurs with the help of bacteriophages. Transfer of genetic material occurs with the help of bacteriophages. First discovered by Zinder and Liderberg. First discovered by Zinder and Liderberg. Okay, in Salmonella typhimurium. Transfer of genetic material occurs with the help of bacteriophages. Okay, discovered by Zinder and Liderberg in Salmonella typhimurium. In Salmonella typhimurium it was discovered. So by these three processes, by transformation or by conjugation or with the help of transduction, the genetic material is transferred from one bacteria to another bacteria. Okay, these are the three methods of we can call it as a sexual reproduction or genetic material transformation. Okay, so that occurs in a bacteria. This is about in detail about the reproduction in a bacteria.